Okay, everyone. I wanted to first thank everyone for attending today's webinar that's going to be covering basic vector construction through vectorbuilder.com. Again, if you have any questions, you can open up the Q&A app and answer or ask a question. We'll answer it live on the air towards the end of this presentation. And also at the end of this presentation, we'll be giving out a promotion code for $50 off your first order on Vector Builder. So let's go ahead and get started. Here we can see the homepage for vectorbuilder.com, where you'll see two primary functions: to design the vector online and to send design requests and other inquiries. If there's anything that you would like to do for your vector that you can't do immediately through our online platform, feel free and click the Send Design Requests and Other Inquiries, and we'll be more than happy to help you with a more customized order. You can also click on this and request modification of an existing backbone should you have a particular vector in-house that you'd like to modify. For today's webinar, we're going to be covering the design of a basic lentiviral vector. And so to get started, what we'll do is click this Design Vector Online button. Once we click the Design button, we'll be greeted with a list of all possible vector backbones. You can see here on the left where it lists lentivirus, adenovirus, piggyback, etc. To the right of that, you'll see an option for a guide. This contains a lot of useful information should you not know a lot about a given vector system. It also includes references to where we have sourced our vectors from. To the right of this, you'll also see a general cost starting from $145, for example, for a lentivirus. This is for a relatively simplified vector design with one open reading frame, one marker, and one promoter. Typically, the price only goes up with custom synthesis. And again, that's based on a per base pair synthesis cost of around 25 to 33 cents. And we'll get more into that in detail later. So for today's webinar, as I mentioned, we'll be starting with a basic lentiviral vector. So here we can see lentivirus, and to the right, we'll see an option that says Go to Design. And again, if you don't have time to design a vector online, you can always send a design request as well. This is available through this option here on the right-hand side, or also through that Send Design Request button on the main page. For now, let's go ahead and click Go to Design. And here you'll see the general interface for all vector design on vectorbuilder.com. On the right-hand side of the screen, you'll see a vector map, which is going to be dynamically updated with any changes that we make to our vector. Similarly, we can also click on any user-generated component to edit that sequence further. To the left, we'll see our selection menu, which will change depending on which portion of the vector we're working on: promoter, marker, open reading frame, etc. First, let's just start by doing a brief overview on some more advanced features. Towards the top here, you'll see number of ORFs. Today, we're just going to be focusing on a one ORF vector design, but should you want to have more than one ORF, you can click this drop-down menu and select up to 4 open reading frames for your vector. Similarly, if you needed to make a large number of vectors, almost nearly identical, but with one component differing, for example, the same open reading frame but different promoters, you can add a component in batch by turning on this option. Well, let's get started by selecting a promoter. On the left here, you'll see a list of our library of promoters organized by category. So we'll start with ubiquitous promoters, tissue-specific promoters, and inducible promoters. Now, if you don't see a promoter that you need, or if you have a custom promoter, you can also click on option 2 to paste a new promoter. If we click that, we'll see that we open up a dialog box to enter that sequence, where you can either enter it by hand or paste it in by using your keyboard shortcut with Control-V or Command-V on a Macintosh. Keeping things simple, we'll use a ubiquitous promoter for this example today, and let's start by just selecting CMV. If we click Select, We can then see that it recognizes that this this step has been completed. And to the right, we can now see that CMV has been added to our vector backbone. As I mentioned previously, if we click on this component on the vector map, it will open an edit window where we can then edit the sequence manually, clear edits that we've made, and display clean sequence if we wanted to copy and paste FASTA sequence into another application. So once we have our promoter selected, we can either click Next Step, or we can click tab number 2 to go to adding the open reading frame. You'll see that we're now on step 2 as indicated here on the left. Also, now that the open reading frame is being highlighted and flashing on the vector map to the right. You'll notice that the interface is nearly identical for selecting your open reading frame from selecting your promoter, except for we now have a new option to select search an ORF database. This actually ties into NCBI's database, and I will get more into that in a moment. You can see here that we have a list of fluorescent reporters as a possible open reading frame, chemiluminescent reporters, chromogenic reporters, drug susceptible markers, regulatory proteins, recombinase, and nucleases. For this example, let's search the open reading frame database. So if we click on this search option, we'll then be brought up with the Search ORF database window. To the left here, we can select a particular species. And to the right, we can enter our gene symbol, gene name, gene ID, RefSeq accession number for both mRNA or protein. For this example, we'll do rhodopsin. So in this case, we'll type in RHO, the gene symbol, and click search. And here we'll see a list of results. To the left, you'll see the organism from which the gene is pulled, along with its gene symbol, full name, aliases, designations, chromosome location, and NCBI gene ID. You'll also see the number of transcripts available to the right-hand side, and an option to view the individual ORF sequences. For results that have multiple pages of results, you'll see you can navigate through each page of result at the top here, and scroll through your results as well. So assuming that we want the human rhodopsin, if we click View ORF Sequences, we'll see a new window with a couple features. First and foremost, we'll see the RefSeq transcripts as identified by their NCBI RefSeq ID and protein ID, along with the size of this open reading frame. We also have an option to edit that sequence or add this sequence directly to our vector. In some cases, you'll also see an additional option of physical resources. This means that we actually have this physical resource as a clone in our facility with an appropriate DNA match of coverage and identity and protein match of coverage and identity. If your open reading frame actually has a physical resource on hand and you select it from this, your turnaround time and cost of your vector will be reduced as opposed to having to perform synthesis to produce your open reading frame for your vector. So in this case, let's select the physical resource since it matches 100% in both protein and DNA for coverage and identity to our NCBI RefSeq ID, and click Add ORF to Vector. We now can see that our rhodopsin gene has been entered into our vector backbone. And again, if we click on this gene on the vector backbone, we'll be greeted with an editing window. Similar to the promoter editing window, we can edit base pairs manually. We can also find open reading frames within the sequence, translate to ensure that our amino acid code is correct, and also add a tag. So for now, let's click on Add Tag, and we can just see general ideas as what tags are available to us, from flag tag, his tag, strep tags, and more. Should you want to add a tag, simply click on the appropriate tag, and you can choose to add it to the N or C terminus. And if we do, we can now see that our changes are highlighted above. But for this example, we won't be adding a tag, so we can just clear our edits. And you'll see that the sequence goes back to the standard open reading frame. So now that we have our promoter and our open reading frame, let's move on to our selection marker. Again, we can move to the next step either by clicking the next tab or by the next step button below. Here we can see that the marker is now being flashed and selected over here on the right-hand side in the vector backbone. And we can see that we now can select from a wide variety of markers here to the left. And again, should you want a more customized marker that's not available on our platform, feel free to design your vector with a placeholder marker and then send a design request to our staff, and we'll be more than happy to work with you. We have an option of drug selectable markers, fluorescent markers, and dual reporter markers as well. In this case, let's go with a neomycin selection cassette. Again, if we select that, we can see that it is now in our vector backbone under its own PGK promoter. So from here, we've designed a very relatively simple vector design. We have one promoter going to one open reading frame with non-modified sequence and one selection cassette. When we're done, we can click Finish, and we'll be taken to our vector report screen. Here, there's a bunch of different options that are available to you. The first off is going to be the cost of your vector here. In this case, because it was relatively simple, we have a price available immediately of $195 with a turnaround time of 10 to 20 days. Should you design a more complex vector, the cost and turnaround time may not be shown, and instead you'll see a green Inquire button. If you click this button, your design will be submitted to our staff for review, and they'll get back to you in 1 to 2 business days with an updated turnaround time and cost estimate. All of our vectors by default are provided in a bacterial stab, and we can see here that this cloning host is the STBL3 strain of E. coli in which the bacterial stab will be provided. However, you can also add a mini prep or maxi prep service to your order as well. Similarly, should you want virus packaging, in this case lentiviral packaging, you'll see 2 options below that for both our standard scale and our large scale packaging. One being 10 to the 8th transfection units per mil, the other 10 to the 9th with a final volume of 1 mil. You'll also see that this adds 6 to 10 days to your turnaround time, and for the mini and maxi prep, 2 days or 3 days respectively. Should you want to go back and make changes to your vector design, you can simply click Back to Edit. If you want to analyze for restriction enzyme digest sites, you can do so with this option as well. Also, one nice feature about the vector report page is that it's fully shareable. The URL that you see at the top of your screen is a permalink, and you can copy and paste it into an email or what have you and share the link with your collaborators. They can then go back and edit the sequence themselves in a non-destructive format, which will generate a new vector ID that they can then share with you or other collaborators as well. You can see here your vector ID. This is what you'll be referencing for your orders, as well as should you have an actual design inquiry. But most importantly, we have our vector report, and we have 2 options, a PDF and a text file. Now, the text file is going to be very similar to NCBI's annotations with various features and sequence detail below. If we go to the PDF format, we'll see our vector report with a full color vector map, vector components annotated by color and description, and a nicely annotated color-coded vector sequence as well. So once we have constructed our vector and are at our vector screen, we can then add this to our cart. Again, if you wish to add a mini or maxi prep or viral packaging, you can do so at this time. Otherwise, you can decline and then go to your cart for checkout. Here you can review your order and all secondary services prior to checking out. And when you check out, you have two primary options. You can purchase now and pay with a major credit card, or you can generate a quote. Once you've generated a quote, this will also assign you a project tracking number, which can then be used in conjunction with your quote ID for placing your order at a later time with a purchase order. To do so, simply go back to vectorbuilder.com, and under the support menu, you'll see the option for placing an order. Here you'll enter that quote ID and project tracking number and upload your PO in a PDF or text format or Word document. You can then enter your billing and shipping information and place your order. At any time, if you'd like to get a hold of us, you can email us at service@vectorbuilder.com or call us at 800-921-8930, and we'll be more than happy to help you with any questions regarding your vector design, placing your order, or the status of your order following placing. We'll be having another couple webinar series in the future going over more advanced vector designs, including editing sequence manually, as well as some of the bioinformatics tools available on the Vector Builder website, including shRNA target design, codon optimization, and more. Similarly, on our products and services page, you'll have an option for positive control plasmids for each vector type, which you can add to your cart as well. At this point, we'll take any questions. And if there are no questions at all, then we would like to invite you to go to vectorbuilder.com. And during the placement of your order, there is an option for a promotional code that you can enter. And simply enter VB Webinar 15. So VB as in Vector Builder, Webinar 15. And that will give you $50 off your order at vectorbuilder.com. We'd like to thank you for attending the webinar today. And again, if you have any questions or special requests, please email us at service@vectorbuilder.com.